Hey everyone, we're looking at the start here of chapter 13, which is all about something that undoubtedly you know a few things about anyway, which is DNA. And so we're going to look at here first in the beginning part of chapter 13, kind of the history behind how scientists identify DNA as being the molecule that is responsible for inheritance and passing genes and traits from parents onto offspring. And then we're going to get into later on in the chapter, the structure of DNA, the sequence of DNA, and how that DNA gets copied during something like synthesis as part of the cell cycle. Okay, so go ahead and get the heading. Again, chapter 13, DNA. So section 13.1 is called Identifying the Substance of the Gene, and you've got a little mouse here. We're going to use mice here as an example of one set of experiments that was done many, many years ago to help identify how traits get passed down and also what is actually responsible for that type of inheritance process, right? What type of molecule there is. And so thinking about the molecules that we've talked about this year, there were four major categories, right? We had carbohydrates, lipids, proteins, and nucleic acids. And among all of those, the most diverse group is going to be proteins. So what that tells us is for a very long time, biologists knew that proteins were going to be super diverse. And so for a while, they were all kind of in agreement that proteins were likely the material that got passed from parents to offspring, and that's what created the enormous variety we see among living things. But as you and I both know at this point, that's not true. We know that DNA is what's actually passed down, and DNA itself is a pretty simple molecule, which makes this a little bit more difficult to kind of comprehend. But we know, and we've known for a century now, that DNA is really what we're looking at when we talk about the inheritance of traits. So the objectives here in the first section, we're going to talk about bacterial transformation in the context of mice, like the picture you just saw. We're talking about viruses specifically as being used in an experiment to help identify DNA as genetic material. And then we're going to talk about a little bit why DNA is so important in terms of heredity. Okay. So when we look at, and I'll move myself over here, when we start talking about DNA and its role in this process of inheritance, the person that we usually start with after Gregor Mendel, because he obviously played a big part in our understanding of probability and the likelihood of traits being passed down. The first person we talk about in terms of like the molecular side of things, right? What's actually causing these traits to appear the way that they are, is a man named Frederick Griffith. His major contribution to what we understand about DNA currently, even though he existed a while back, right? He's not still living. He identified what we called a transforming principle. Now, that is what we've come to know as DNA. That is what we've come to know as genes and chromosomes. But at his time, we had no idea about what those things were. Okay. So he identifies this thing that he calls a transforming principle. And that kind of sets off like a firestorm historically, where lots of different scientists are trying to then take his work and add to it, right? What is this transforming principle? So the way that he identifies this, the research that he did in order to find this transforming principle, was done with something called bacterial transformation. So transformation is a vocab term. You can see it's in bold there. You should definitely write that down. This is the permanent genetic alteration of a living thing, such that it shows characteristics of something else. Scientists do this in the lab all the time, and they generally do it with bacteria, which is why I said bacterial transformation. What it essentially involves is taking genes, genetic material from one organism, putting them into bacterial cells, and then the bacteria in response to that will express those traits. Okay. So Griffith knew that this was a process that could happen, but he didn't really understand how that worked. So he designed an experiment that incorporated mice and infectious bacteria, specifically the bacteria that causes pneumonia. He used two forms of this bacteria, the S form and the R form. The S form is deadly. The R form is not so deadly. These were two forms that were identified a long time ago in the medical literature for people that were actually studying pneumonia and its effects on humans. So he's using the deadly form and the not so deadly form. And by transforming and using these bacteria to kind of infect mice, he was able to see exactly what was happening in terms of passing on maybe these more deadly forms. So at the bottom here, it says results showed that transforming material passed from heat-killed or dead S bacteria to live, causing the R to transform and become deadly. So I'm going to show you here on the next slide what that's actually referring to, and then I'll step back here and reiterate that one more time. So what we have here is the setup for Frederick Griffith's experiments. So he's got a petri dish here, and we have two different types of pneumonia-causing bacteria. So the S type is here. S type causes disease, and S type also generally causes death. And so you can see if you just inject a mouse with bacteria that is the S type, the more dangerous form, then the mouse will eventually die of pneumonia, as expected. Okay. However, if you use this R-type pneumonia-causing bacteria, we don't actually get an active infection. The mouse survives, and it is just generally healthy in that case. And so we see kind of the opposite effect here with that one. And these are what he would call controls, right? These are the things that he would expect to happen. Now, where his actual experiment came in to identify this transforming principle is in these two right here. So this one in the third panel and in the fourth. So the first thing he did then is he took S-type bacteria and he basically heated them up in this little test tube right here. And what that does to any living thing, if you heat it up enough, it's going to cause the cells to rupture. Some of the molecules may start to fall apart. But in general, what that means is that if it's something that's infectious, it's probably not going to be able to cause an infection later because it can't just kind of reassemble when it cools down. So if you heat kill that S-type bacteria and then you inject it into a mouse, the mouse lives. And that's pretty much something that was, well understood at the time because you've essentially kind of shredded that dangerous bacteria apart in a way that it can't reassemble and it can't infect anything and cause harm. And so that was something he expected. Now, where something kind of new happened, where he was able to figure out that there was something that was being passed and is used in this transformation process, was he would take these heat-killed S-type bacteria that were normally harmful, right? But in this context, when they're injecting the mouse still lives. And then he combined that with the harmless R-type bacteria. And what he found was really kind of interesting. When he injected that into the mouse actually ended up dying of pneumonia, which was like crazy unexpected for him. And it's still probably kind of hard to comprehend because what we're actually talking about here is that this information, the genetic information that was found here in this S-type bacteria that was killed by heat, where all that stuff is kind of shredded and broken apart, that stuff actually got reassembled into these healthy, harmless R-type bacterial cells. So they actually kind of took in those genes and the other proteins that were maybe present in that heat-killed bacteria originally, absorbed those into their cells, and then expressed them in a way that caused the mouse to actually die. It caused disease, it caused pneumonia, and the mouse died as a result. So he, again, had no concept of what that is. We now know that we can say, this is genetic base, it's genes, it's proteins, it's things that we're inheriting, right, and being passed down. But in terms of what Griffith was doing, he didn't know what those molecules actually were. He was kind of looking at all this on the surface level, right? So this is where he says, okay, there's this thing called a transforming principle. It got transferred from my heat-killed S bacteria to my harmless R-type bacteria and then killed the mice. And so this is where that conclusion came in, where I was on the last slide right here. So transforming material gets passed from heat-killed or dead S bacteria to live R, causing the R bacteria to transform, right, to start looking and acting differently. It's a permanent genetic change, and then therefore they became deadly. All right, so he did his work again and published his data. And following that, there were a whole bunch of other scientists that came along that were also kind of like actively trying to investigate, what is it that is actually transmitted? What gets passed from parents to offspring? What kind of molecule is that in a cell? And based on Griffith's work, they all kind of jumped on that bandwagon and they said, okay, we're going to figure out what this is if we can identify what this transforming principle is. So there was a whole group of scientists, but the one that we tend to talk about the most is a guy by the name of Oswald Avery. And Avery was in communication with Frederick Griffith. And as part of that communication, he was able to isolate and purify his transforming principles. So he actually took Griffith's data, Griffith's results, the molecules that were involved in his study, and he was able to isolate the molecule that he thought was responsible for transforming these harmless bacteria into harmful bacteria. Okay, so again, the thing that was being passed from one to the next. So what he identifies as part of that, he gathers all sorts of different types of data. He did qualitative analyses. He did analyses of enzyme activity. He looked at the chemical makeup of that isolated transforming principle. And all of those signs, all the results that he got, all the data he gathered from all of that tended to point to this idea that it was likely going to be DNA that actually made up this transforming principle, which was at the time like totally unheard of. This was not a concept that anyone would have really thought to be true. Again, everybody was kind of in that train of thought where they were thinking that it was going to be protein. It had to be protein because proteins are so diverse. DNA is just not. DNA is a very simple molecule. And so for Avery and his group to come along and say, hey, this looks like DNA, there wasn't a ton of support for that. All right. And so what's going to happen as a result of that is there are going to be additional scientists that kind of replicate his work to make sure what he did was correct and valid. And then there's going to be some that kind of extend upon that and add to our understanding of this transforming principle. Okay, but the statement up there at the top, scientists discovered that nucleic acid DNA stores and transmits genetic information from one generation to the next. So DNA in this case, based on Avery's work, was identified as a transforming principle. So when we look at this table here specifically, all right, this is looking at Avery's research in terms of the chemical makeup. So this is a quantitative type of assessment. What matters in this case, there's nitrogen that he's quantifying. He's trying to see the percentage of nitrogen present in these different samples of the transforming principle. And then he's looking at the percentage of phosphorus in these samples also. Now, there is a known value for DNA, and that's consistent with what we know about DNA right now. DNA contains nitrogen and phosphorus in a ratio of about 1.5 to 1, 1.69 to 1. So in general, for every one phosphorus atom that we see 1.5 to 2 nitrogens, right? Somewhere in that range. So it's not, exactly at one number, but that's kind of our average for any kind of sample of DNA. There was a quantification of the amount of nitrogen and phosphorus that had also been done at the time for other types of molecules, including proteins. And in those assessments, we get very, very different numbers. Okay. It is not 1.5, 1.69 to 1. It's going to be something that is much, much lower. Okay. In that case. So that's kind of what we're looking at here. All of these samples, the percentages of nitrogen, you can see they all kind of fall in line with what we know for DNA. Percentages of phosphorus, again, fall in line to what we know for DNA. And the ratio here is going to be very similar also. Okay. When he used enzymes that break apart proteins, nothing happened in his test tubes. But if he used enzymes that were known to break apart DNA, everything kind of fell apart. So lots of, again, research went into this. Many, many years of studying these in different contexts was helping to kind of identify DNA as this transforming principle. The very last pair of scientists that we're going to talk about in this first section here, these are not going to be your last scientists for the chapter, but at least in 13.1, there were two scientists. This is Alfred Hershey and Martha Chase, and these were virologists. So that means they are scientists that study viruses, and specifically they were looking at viruses that infect bacteria, which kind of seems a little strange, I think, if you think about it. Viruses that infect bacteria, first of all, who cares? And second of all, why? What's the point in that case, right? Viruses, are known to infect lots of different types of living things, but bacteria are not one that we tend to think about very often with this, right? But as part of their study, they have an idea of what's going on with this transforming principle research. They knew of Avery and his group's research. And so they wanted to extend and build upon that and see if they could also kind of confirm what Avery had said and what most people at the time, again, were still not believing to be true. And so what they did with their bacteriophages is they were able to take that virus and tag that viral DNA with radioactive phosphorus. Okay, and then they're going to tag proteins with radioactive sulfur. So proteins contain sulfur, DNA contains phosphorus. All right, DNA definitely does not contain any sulfur. So that essentially means if you can distinguish between the radioactive phosphorus and the radioactive sulfur, you can identify, what's being used to kind of infect bacterial cells on the part of these viruses. So again, if you remember from the start of the year, the way viruses work, they're essentially protein shells on the outside. And then on the interior here, they have DNA. It can be single-stranded DNA, double-stranded DNA, single-stranded RNA, or double-stranded RNA. We'll talk about RNA in chapter 14. But how they infect is they attach to the cell membrane. So this greenish, bluish part on the outside would be the cell membrane. And they inject that genetic information into the cell. Okay, and in much the same way that we pass genes from one individual or organism to another, right? We're combining them in different ways. Viral DNA does the same thing, and it does the same when we talk about a virus and bacteria. So the virus normally will inject its DNA. If we have tagged viral DNA that's been kind of tagged with this radioactive phosphorus, which you would expect, if DNA is actually being used as this infectious process or this pathway, you would expect to see radioactive phosphorus here inside the cell. Okay, if DNA is not being used and the virus is actually using proteins, then you would expect to see radioactive sulfur on the inside. Okay, and so what they did is they set up an experiment where they kind of combined these things separately, and then they essentially took them and they threw them into a blender, like legitimately threw them into a blender and spun them really fast. What you end up getting is you get a pellet at the bottom that contains the solid parts, and the liquid at the top is much less dense, right? Like with anything, if you're going to separate it density-wise, the more dense thing goes to the bottom and the less dense is up at the top. And so when they assessed, what was present in the cell material, they were able to identify, where the radioactivity was. So this is essentially what their experiment looked like. So they had, again, bacteriophage. One of them is tagged with a radioactive DNA. That's phosphorus 32. That's just the radioactive one. Here we have the other type of virus that's tagged with the radioactive protein or sulfur. When those are allowed to attack bacteria, you get one of two options, right? Either the radioactivity goes inside the bacteria if it's DNA, or if it's protein, the protein radioactivity will go inside. But what they noticed was that anytime they ran this trial up here, the one up at the top with the radioactive DNA, they always saw radioactivity inside the cell, meaning the DNA seemed to be going into the cell. When they ran this trial down here, where you have the radioactive proteins, they never found proteins in the cell materials. They only found proteins in kind of the liquefied parts of their little test tubes that they spun around enough in the blender in order to separate the solid stuff from the more liquid stuff. Okay, so the radioactivity remained separated from the cell when it was tagged on the protein. All right, so scooting back for a second, their conclusion was this part down here. So they found tagged DNA was found inside the bacteria, but proteins were not, showing that only DNA could actually be genetic material. It was what viruses use in order to infect their host. It is what we saw in the transforming principle with bacteria, and it's likely also going to be the same in the remainder, all the other living things on the planet. And we know now, certainly with the technology we have, that these claims were correct, right? So DNA is what we inherit, not proteins, but proteins get made from the genes that we inherit from our parents in the form of DNA, okay? So when we talk about DNA as genetic material, right, as that material that we use in terms of inheritance, it has several different roles. So it stores genetic information, right? There are some things that we inherit, like a carrier, for example, that doesn't get expressed, right? But it can also store lots of other information that can then be passed on to our offspring or onto new cells that need to be created through mitosis. It also can be used, and it provides a good template to make a copy of itself, which is helpful, again, with things like mitosis and meiosis when we go through that cell cycle, but also important for fixing damage and correcting errors. And then express. So we use genes and DNA, the instructions, the code for our proteins to actually show a trait, like a phenotype, okay? So DNA is going to be, again, the focus of this chapter. We're going to come in and talk about the structure of DNA here in the next section. All right, so that's all I've got for today. If you've got any questions, as always, let me know. Otherwise, I will see you later. Bye.